Good morning, students. I hope you are staying at home uh, and taking proper precaution to avoid the coronavirus. Today, we are going to discuss about one of the important public health problem for the mostly for the developing country, and that is enteric fever. The name, as it you understand, the name itself says that this has something to do with the intestinal system or gastrointestinal system. That means the organism enters through fecal-oral route. What is the purpose of the what are the objectives? The objectives should be that at the end of the lecture you should have a clear idea about the etiological agents, the epidemiology involved worldwide, Indian scenario, how the organism invades and what kind of immune invasion it produces and how does it produce the disease, and we will talk about the laboratory diagnosis of this particular disease, that is enteric fever, typhoid both as well as paratyphoid fever, and we discuss about the antimicrobial, the relation to the drug resistance, as well as talk about the vaccine, because now we have an excellent vaccine for typhoid fever. With this objective, let's start. The disease caused that we call enteric fever is caused by an organism called Salmonella. Well, Salmonella causes two types of infection. One is causes invasive disease, where the organism gets into the blood system, causes septicemia, causes multiple organ involvement, and on the other side, it can stay locally on the gastrointestinal mucosa and produce severe acute gastroenteritis, which may be self-limiting, usually self-limiting. So these two groups of salmonella are called typhoidal salmonella, which includes salmonella typhi as well as salmonella paratyphi A, B, and C. Most of the time in India, as you will realize, we are talking the enteric fever or typhoid fever is caused by Salmonella typhi and Salmonella paratyphi. Whereas non-typhoidal salmonella are, as I already told you, that non-typhoidal salmonella causes mostly self-limited gastroenteritis. But at some point of time, when the immune system is not is compromised, or in very small children, even non-typhoidal salmonella can enter the bloodstream. But that's very rare, mostly seen in sub-Saharan Africa. So with this understanding that Salmonella is a gram-negative organism, it belongs to the Enterobacteriaceae family. As you all understand that Enterobacteriaceae family is a heterogeneous group of organisms that shares certain common phenotypic as well as genotyping characteristics. As I already told you that the enteric fever, the major etiological agent of the enteric fever or typhoid fever is Salmonella enterica serotype typhi, or which causes typhoid fever. But a significant number of typhoid fever can also be caused by Salmonella enterica serotype paratyphi A, which we call paratyphoid fever. In fact, if you look at the epidemiology number of cases, in recent year, even the number of the paratyphoid fever has increased. That they also pose a problem as problematic as typhoid fever, because they also carry a large amount of antibiotic resistance property. The other Salmonella, serotype paratyphi B and paratyphi C, are mostly not seen in India. These are mostly in the developed country like US or South America, whereas paratyphi C is mostly seen in the Middle East. As with all the gram-negative organisms like E. coli, this organism also classified into different serotypes based on the property of the antigen on the surface of the bacteria. As you understand that a gram-negative organism has O antigen. It has O antigen is a part of the lipopolysaccharide that extends from the outer membrane, and it has also something called a flagellar antigen or H antigen. It has, and this organism, specifically Salmonella typhi, has a capsule, polysaccharide capsule on the outer surface of the organism called VI antigen. This is a polysaccharide antigen. VI stands for virulence, virulence in mice. The other factor you have to understand is based on the ONH antigen, a huge number of different monoclonal or polyclonal absorbent sera has been created or has been made, and this can be utilized to type this gram-negative bacteria or Salmonella typhi into more than 2000 different serotypes. The other facts about the ON and the H antigen is that antibodies raised against this O antigen and H antigen can be utilized as a diagnostic marker, or this antigen can be used as a diagnostic marker in one of the oldest tests that is used for enteric fever is called Widal test. So O antigen, we look for antibody to O antigen, and H antigen we look for anti-H antibody. Okay. So if you look at the epidemiology of the disease, first we have to understand is that there is a marked difference between these two organisms, both the Salmonella typhi and the typhoidal Salmonella and the non-typhoidal Salmonella. I have already told you that non-typhoidal Salmonella causes acute gastroenteritis, and that is self-limiting disease. The non-typhoidal Salmonella is mostly transmitted through it is seen in large numbers in different kind of animal, non-typhoidal Salmonella like Salmonella enteritidis, cholera suis. These are found in the guts of the different kind of animals, poultry, cattle, dogs, maybe sheep, even reptiles like snakes, or it can be also found in the turtles, but these are non-typhoidal salmonella. So non-typhoidal salmonella can be transmitted through eggs, or it can be transmitted through meat products, or it can be transmitted through some food product contaminated with feces or contaminated vegetables. So there is a difference between the host specificity of the organism. As I told you, non-typhoidal salmonella can infect a large number of different species. It can infect cattle, it infect poultry, but whereas Salmonella typhi and Salmonella typhi are absolutely restricted to only humans. So there is a gross difference between these two. So the humans are the only source of infection as far as the enteric fever is concerned. Salmonella typhi and Salmonella typhi A, B, C all are found in human. Why is this specificity of Salmonella causing enteric fever? When they looked at the genomes of these two organisms, non-typhoidal Salmonella and Salmonella causing enteric fever, they found that in non-typhoidal Salmonella or typhoidal Salmonella, there are about, out of the 4,000 genes that have been studied in Salmonella as a whole, about 200 genes are non-functional in typhoidal Salmonella. That means because of the loss of function of these 200 odd genes, it has lost the specificity to adapt to a large variety of different organisms. So this is very enteric or typhoidal Salmonella are very specific to human. It doesn't survive in any of the other species. Then we have to understand also what are the risk factors. As I told you that this is it. The typhoid. We are going to discuss only typhoidal Salmonella. Non-typhoidal Salmonella or acute gastroenteritis will be separately discussed, or all had probably already been discussed as a cause of acute gastroenteritis or food poisoning. So today's discussion will be only specifically restricted to enteric fever. As we understand that this disease is fecal-oral transmission, that means it is associated with food, it is associated with water, it is associated with any kind of material that we consume, and there is a possibility that this can be contaminated with some typhi. And this is mostly in if personal hygiene is not appropriate, if good quality drinking water is not available, if public is not aware of the disease, if there is lack of education, if there is a large crowding as in we seen in the developing country. These are all factors that helps the spread of the disease. So as we understand, this is a disease of the developing country. In developed country, because there is a higher standard of personal hygiene, higher standard of good quality drinking water, public awareness, less of crowding, enteric fever is not normally is frequently seen as it is seen in the developing country. As I already told you that paratyphoid fever caused by paratype B as well as paratype C are mostly not seen in India. B is mostly seen in Europe and Britain, whereas paratype C is mostly seen in the Middle East. I already mentioned that typhoid fever is a major public health problem because it is a disease of the low socioeconomic condition. If you look at the overall the morbidity and mortality associated with disease in a study carried out in the Lancet in 2019, it was found that in 2017 there are about almost 11 million cases were reported, and about 117,000 death has been described. But from 1991 to 2017, it has been found that there has been a drastic drop in number of cases of typhoid fever, about 53% drop in the number of cases, as well as 50%, 58% drop in deaths. Why is the difference? In if you compare it from 1990 till recent time, this drop is basically because of the better antibiotic availability, is because of better drinking water, it is because of better personal hygiene, is better because of the better education system, public awareness, and it's better because there is a good vaccine available now. Similarly, paratyphoid fever that has been reported in 2007 about 3.4 million, and that's about 19,000. So, and if you compare the paratyphoid way back in 1990 and as of now in recent years, there is again drop of both the cases as well as this is a good sign. And as I have already mentioned, that this is because of the drop in because of good hygiene, availability of good drinking water, because of better management, newer antibiotics, as well as the appropriate vaccine that is available, effective vaccine that is available. Let's quickly look at the epidemiology of the disease, both and typhoid as well as paratyphoid fever. If you look at the first graph on the left side, it says global age-specific incidences rated per 100,000 population. So if you look at the age group, age in this is in the x-axis, it is the age of the child. On the y-axis is the incidence per 100,000. If you look carefully, the majority of the incidence of the typhoid fever is in the younger age group, mostly below 15 years. The maximum majority of the cases occurs below 15 years of age. Similarly, the mortality rate also is very high in the younger age group, and this is more true for the developing country. So typhoid incidence, if you look at the number of cases that occurs worldwide, this is data from 2015, but this is still very true for as of now, and you can see the countries that are mapped in red color has the highest number of cases in the world. In India, the number of cases varies anywhere between 450 to 500 per 100,000 population. So India, the Southeast Asia, Indonesia, part of the South East Asia, as well as most of the impoverished African countries, you have a very high incidence of typhoid fever. What is the burden of in India? As of 2019, published in AIIMS, there has been about 6.6 million cases in the year 2016, and there are about 66,500 cases. That was, most of these cases were below 50. So for India, this is a major public health problem. The mortality rate, as you will see, the mortality rate due to enteric fever is mostly in the developing country, and unfortunately India has the highest number of mortality in terms of per 100,000 population. As you see, it's about 100, between 50 to 80 per 100,000 population. So this is a public health problem associated mostly in the developing country. Like any other gram-negative organism, this organism, as I told you, is invasive organism. Salmonella is an invasive organism, therefore it has certain amount of virulence factors like the invasive E. coli or enterotoxicity E. coli. So first thing is that it has a large number of different adenosine molecules, which we are going to discuss in a little while. It has the ability to survive inside the epithelial cell, inside the phagocytes, inside the dendritic cell, even inside the B cell, and then it can multiply inside these cells and survive for a long period of time. It is the endotoxin or lipopolysaccharide that is a part of the gram-negative organism is also an important virulence factor. In addition, it produces a large number of different enterotoxins. This holotoxin of E. coli that we have described earlier is also quite frequently seen in typhoid fever. It also produces cytotoxin, vacuolating cytotoxin-like structure, which induces pyroptosis, and it can kill the epithelial cell. Thereby, it can cause perforation in the intestinal epithelial cell. The other important thing about the virulence of this organism that Salmonella, in addition to its own host chromosome, it carries extra chromosomal component like the plasmids, transposons, or it can carry a pathogenicity island. In fact, for Salmonella typhi, the pathogenicity islands are the most important virulence factor that we are going to discuss in a little while. VI antigen, as I already mentioned, specifically for Salmonella, is an important component, and this VI antigen production is encoded also in the pathogenicity island. The other thing you have to remember is that pathogenicity island is a transposable element, but it can be a stable part of the host chromosome. So the pathogenicity island gets integrated into the bacterial host chromosome, whereas the plasmid can be lost or plasmid can be acquired. So this organism has both the plasmid as well as the pathogenicity island as a transposable element carrying a large number of different properties. This can be more clear if you draw a comparison between Salmonella typhimurium and Salmonella typhi. In Salmonella typhimurium, I already told you this is a non-invasive organism, or it is locally invasive, stays in the intestinal mucosa. It doesn't spread to the different parts of the body or doesn't get transmitted into the blood. Whereas the enteric Salmonella or typhoidal Salmonella has the similar kind of structure: inner cell membrane, peptidoglycan, outer cell membrane, plasmids, VI antigen specifically for Salmonella typhi. So this organism, as I told you, pathogenicity islands that are present inside the Salmonella typhi is different than the pathogenicity island that is found in the Salmonella typhimurium. So majority of the virulence factors that are associated with invasiveness of Salmonella typhi as well as paratyphi and paratyphi B are because of the pathogenicity island which are found inside the salmonella chromosome, and these are very specific to salmonella typhi, and these are not found in salmonella embryo. The other thing, important thing you have to understand is that Salmonella typhi also carries something called a type 3 secretion system. This is a very important virulence factor because this type 3 secretion system, it is able to inject effector molecules produced inside the cytoplasm of the bacteria into the host cells. That we will see in a little while. Let's go back to the first lecture we had on the bacterial genetics. We have to understand because a majority of the virulence factor in case of typhoid fever in the Salmonella typhi organism or paratyphi or paratyphi B is encoded in the pathogenicity. What are pathogenicity islands? Basically, pathogenicity islands are huge chunks of DNA that has the bacteria has acquired, salmonella bacteria has acquired, typhoidal salmonella bacteria has acquired over a period of evolution, maybe millions of years. This genetic component, which can, as I told you, which can be maybe 50 to 150 kilobases long, carries a large number of different properties. But how do you know? And as I told you, this pathogenicity island is inserted into the host chromosome of the bacteria. It doesn't have a independent existence like the plasmid. It is a part of the host chromosome. So how do you know the pathogenicity island that exists inside the Salmonella typhi? There are very specific markers that you can identify pathogenicity island inside the Salmonella typhi. First is that the G plus C content of the guanosine and cytosine component in this fragment is much different than what you find in the rest of the chromosome. Suppose in this particular strand of DNA, the G plus C content is about say 56%, whereas in the rest of the DNA this can be 46%. Just give you an example. So first point to identify a pathogenicity island in Salmonella typhi is their G plus C content. Secondly, if one looks at the genome of the Salmonella typhi, one should be able to identify insertion sequences at both the ends of this particular segment of DNA, and also it has a direct there is a indirect repeats at the both inverted repeats at the both the end of the DNA, plus the part where this particular pathogen cell is inserted, which we call the target DNA in the host chromosome, will be duplicated. The same copy of the gene will be present in the both sides. This is part of the original DNA of the bacteria. This is a foreign DNA which is a part of the pathogenicity island. And this pathogenicity island carries a huge number of different virulence property that helps the organism to invade inside the cell, survive inside the cell, and gives property to make it less immunogenic, or the gives ability to the bacteria to manipulate the immune system. And in Salmonella typhi, there has been more than 21 different pathogenicity island that has been described. Salmonella typhi, typhoidal salmonella, there were more than 21 different typhoidal pathogenic island that has been described. So what are the function of this? Just give an example. What are the different function of the pathogenicity island? Pathogenicity island can help invasion. It helps the organism to survive intracellularly. The pathogenicity island gives the organism to produce VI capsular polysaccharide, and it helps the organism to inhibit phagolysosomal fusion inside the bacteria. This pathogenicity island, just like the plasmid, also carries large number of different antibiotic resistance, like extended resistance to extended spectrum beta lactamase. Just to give an example, how this pathogenicity island inside the chromosome of the bacteria. This is a plasmid. This can be the DNA of the bacteria, and this is a pathogenicity island number 1. As I told you, there are 21 different pathogenicity islands, but most of the virulence factors that are associated with invasiveness are associated with 4 or 5 or maybe 6 different type of pathogenicity island. So this pathogenicity island produces a large number of different components of virulence factor outer membrane protein specifically that helps to bind it to the epithelial cell. For example, SHDA binds to fibronectin, MISL helps to again bind to the fibronectin, STIV, salmonella typhi invasive protein helps to bind it to the epithelial cell. The VI capsular polysaccharide inhibits complement deposition on the surface, etc., etc. As I told you, the salmonella typhi pathogenicity island confers the organism a large number of different properties. One of the important property is the type 3 secretion system with injection system. So the organism first gets attached to this is intestinal epithelial cell. First the organism gets attached to the intestinal epithelial cell, then it injects an effector molecule inside the epithelial cell through the type 3 secretion system and creates a salmonella containing vacuoles. It's called SCV, salmonella containing vacuoles. This entry can not necessarily through the type 3 secretion system only, but it can be also type 3 independent type 3 secretion system independent system. That means the organism, in addition to this, the type 3 secretion system has also other virulence marker that helps the organism directly invade into the cytoplasm and create a salmonella containing vacuoles. Now once the salmonella is inside the salmonella containing vacuoles in the epithelial cell, it has two different parts. Either it can break it, the salmonella containing vacuole, and get into the cytoplasm, or it multiplies inside the vacuoles in large numbers, and this will require additional virulence effector molecules that are also present in the inside the host inside the bacteria. So at one point of time, when the bacteria produces a large number, it will break out the salmonella containing vacuole, induces apoptosis inside the cell and or pyroptosis and through the inflammasome, and this pyroptosis will lyse the cell and the bacteria will be released. So what does, I mean, if you look at the pathogenesis, let's quickly look at the pathogenesis of how this organism interact with the host immune system. If you look at the host immune system in the intestinal epithelium, you have to understand that there are two important factors that are necessary for innate immune response. One is that the epithelial cells, or most of the immune cell, has the ability to recognize pathogen associated molecular patterns. So the organism carries different kind of structures on its surface, like the lipopolysaccharide, like the flagellin, like the outer membrane protein, like the DNA it carries, like the cytoplasm it carries, and these all are called pathogen associated molecular pattern. So once the organism is on the attached to the surface of the epithelial cell, on the epithelial cell there are a large number of innate immune receptors which are called toll-like receptors or nod-like receptor or nucleotide polymerization domain receptors. These toll-like innate immune receptors of the immune cell can be present on the surface of the epithelial cell or monocyte macrophages. It can be also present in the cytosol of the epithelial cell. So once the bacteria comes in contact with the pathogen-associated molecular, once the host cell comes in contact with the pathogen-associated molecular pattern, and this innate immune receptor on the host cell or intestinal epithelial cell induce myelin dependent D88 cascade system, and through the NF-kappa system it will produce the pro-inflammatory cytokine like IL-1, beta, TNF alpha, interferon gamma, interleukin 18, interleukin 8, so on and so forth. So this epithelial cell has all the capacity to produce a large number of different inflammatory cells, and these inflammatory cells will these inflammatory cytokines will diffuse into the subepithelial or submucosal or the basolateral aspect of the epithelial cell, and it will it will activate the different macrophage monocyte that is present on the basolateral aspect that we're going to discuss. The other thing it does is the bacteria does is the there are there there are another there is another term called danger associated molecular pattern. This danger associated molecular pattern are endogenous factors that are found in the host epithelial cell. These two components, pathogen associated molecular structures from the bacteria and danger associated molecular pattern, these are present inside the host cell. So once this pathogen associated molecular pattern is recognized by the toll-like receptor, the inflammasomes are produced inside the cytoplasm. What are these inflammasomes? These inflammasomes are basically a collection of innate immune response structure which has the capacity to produce inflammasome. The function of this inflammasome is that it will induce production of the inflammatory cytokine. Also, it will induce caspase activity. What is the function of the caspase activity? That it will cause lysis of the induced apoptosis or pyroptosis inside the epithelial cell, and it will cause lysis. Once the cell dies, the cytokine produced, it diffuses into intestinal epithelium, and subsequently it will produce inflammatory sign. But specifically for Salmonella typhi, Salmonella typhi is somehow downregulates the inflammatory response inside the epithelial cell. So, unlike the Salmonella enteritidis or non-typhoidal Salmonella, where it produces large amount of inflammatory response, typhoidal Salmonella somehow manipulates both the epithelial cell as well as the macrophages. I'll just come back in a moment. So once the Salmonella enters the epithelial, so through 3 AIIMS cells, and subsequently it will come in contact with the macrophages or the T cell and B cell, and it will produce and the cytokine will induce a certain kind of inflammatory response. But typhoidal Salmonella somehow manipulates both the epithelial cell as well as the macrophages and the T and B cell, and it suppresses the immune response in the intestine. Why does it do that? If it is able to suppress the immune response inside the epithelial cell, then it is able to survive inside the epithelial cell, and quietly it can disseminate to different parts of the body. Whereas non-typhoidal Salmonella will produce a huge amount of inflammatory response here, and this inflammatory response will attract large amount of polymorphonuclear leukocyte, monocyte macrophages, and they will come and kill the bacteria at over a period of time. Therefore, non-typhoidal Salmonella is self-limiting, whereas typhoidal Salmonella, because it is able to manipulate the epithelial cell, it's able to manipulate the T cell, it manipulate the B cell, manipulate the macrophages, it is able to reduce the amount of immune response in the intestinal epithelium, and therefore if there is not much inflammatory response in the submucosa, then it is able to disseminate into the different parts of the body through the inside the macrophages. So coming back to the virulence factor, as I told you, just to give an example, that the pathogenicity island, Salmonella pathogenicity island, I told you there are 21 of them, but majority of these factors, the factor molecules that are inserted inside the epithelial cell or monocyte macrophages are carried in the Salmonella pathogenicity island 1 to 7. So these, what for example, what do they do? They will cause maturation of the salmonella containing vacuoles. It will help invade inside the epithelial cell. It will help survive the intracellular survival of the organism. So this pathogenicity island produces a large number of different effector molecules that helps it to survive inside the epithelial cell. So what are the, for example, what are the different properties encoded by this pathogenicity island? For example, pathogenicity island 1. Salmonella pathogenicity island produces something called SIIE, Salmonella invasive I protein E. This is a non-fibrillar adenosine. It helps to attach to the epithelial cell. The Salmonella typhi also produces biofilm associated protein or called BAP. It is on the bacterial surface, helps it to attach to the epithelial cell. It has something called RCK or resistance to complement killing. It is a part of the outer membrane protein. Similarly, there is something called a PAG9 outer membrane protein, helps to the bacteria to attach to the heparin sulfate on the host epithelial cell, or something called Salmonella typhi invasion is a part of the outer membrane protein, helps it to attach to the epithelial cell again, and the other outer membrane protein which is called MIS or misal is a part of the outer membrane protein and helps to adhere to. So there is a large number of different adenosine proteins which are encoded in the pathogenicity island, and this helps the organism to invade into the cell after it attaches. Okay, so let's quickly look at the pathogenesis. As I told you, this is transmitted through the fecal-oral route. It can be through food or it can be through water. And once the infective dose is pretty large, it's about 10 to the power 7 organism per ml. So it's a huge amount of bacteria. Otherwise, it will not be able to survive the acidic pH or the alkaline pH, acidic pH of the gastric juice, because the gastric pH can be as low as 2. So to survive, it requires a large number of organisms. So after ingestion and crossing the epithelial layer of the intestine, as I already am going to just show you in a little while, it will transmit to the blood and cause bacteremia. So after 7 to 14 days of incubation, it is already adhered to the intestinal mucosa, and then in the submucosa it will encounter dendritic cell. In the Peyer's patches, enterocyte, macrophages, the gall associated, these are all part of the gut associated lymphoid tissue, and over a period of time this organism will carry it from the Peyer's species mucosal associated lymphoid tissue to the mesenteric lymph node, and in the mesenteric lymph node these are carried inside the macrophages, inside the dendritic cell, and this will go to the reticuloendothelial cell, mostly the liver and the spleen. From the liver and spleen, it will be again the cells carrying the monocyte macrophage dendritic cell carrying the bacteria resides in the liver and spleen, and from the liver and spleen it will inside the cell it will multiply and get disseminated into the blood, and from the blood it can transmit to bone marrow, it can transmit to liver, it also transmit to the gallbladder, and from the gallbladder it can again come back to the intestine through the secretion and reinfect the individual. And if the individual is not treated and still suffering from the bacteremia at the end of 3 or 4 weeks, then it will have severe complications like the encephalitis. It can have necrosis of perispaces leading to perforation, peritonitis. It can have myocarditis. It can have a large number of different complications leading to mortality or leading to death of the individual. This part I already told you. The intestine, you have the M cells. These are membranous cells found in the intestine. These are antigen processing or antigen absorbing cell in the intestine. These are not cells which are associated with absorption of the nutrition. The bacteria probably enters through the M cell, probably it also enters through the normal epithelial cell. As I told you, just below the mucosa, you have a large amount of gut-associated lymphoid tissue. This gut-associated lymphoid tissue will have macrophages, and these macrophages will send out their pseudopodial structure to the outside of the intestinal epithelium, and it will grab the bacteria, and then it can either phagocyte the bacteria, kill it, or if the bacteria is very virulent, then it will able to survive inside the macrophages, and this macrophages then will be transported to the reticuloendothelial cell, specifically spleen, where this may be processed and presented to the T and B cells. Some of these T and B cells, B cells specifically, will also be invaded by the bacteria. So once in this state, carried by the macrophages, inside the macrophages infected with the salmonella bacteria is able to survive inside the salmonella containing vacuoles. The effector molecule of the bacteria will inhibit phagolysosome formation, and the bacteria will multiply in large number, and it will be carried into the different parts like the spleen, or it will be carried to the liver, it will be carried to the bone marrow, or it can to the gallbladder, where it can survive. If there is a stone in the gallbladder, the organism will produce a biofilm, and it will survive for a long period of time. And or from time to time from the gallbladder, it can be disseminated. What is the clinical picture? Mostly the disease presents with fever, abdominal discomfort, but over a period of time there may the pathosplenomegaly, as I told you, because the reticuloendothelial system has been invaded, so the spleen will react, the liver will react, and it will become enlarged. Gastrointestinal system may be not usually present in the first week, except abdominal discomfort, but over a period of time the intestine may be large amount of disruption will lead to severe, it can lead to diarrhea or severe abdominal pain, or it can lead to perforation and peritonitis. And even if this point of time the infection is not interrupted with the appropriate antibiotic, the organism can to travel to different parts of the body, like example CNS. It can be found, it can cause encephalitis, and it can be found in the CSF. The bacteria can be found in the CSF. In fact, in the Southeast Asia, toxic encephalopathy is a major issue. It can also produce myocarditis, septicemia, and leading to septic shock. This I already mentioned that the organism can cause gastrointestinal bleeding, perforation, peritonitis, encephalopathy, hepatitis, toxic activity, etc. The other thing, important thing that you have to understand is that it creates a carrier stage, and the carrier stage is mostly, as I told you, because the organism is able to survive inside the gallbladder for long period of time. So what are the different carrier stages? There is a definition called temporary carrier. This temporary carrier may be the organism may be inside the intestine, and if the organism is shaded for more than 3 months, up to 3 months, this is called temporary carrier. That means at the end of 3 months the individual is able to somehow eradicate the bacteria, and the temporary carrier stops. But in chronic carrier, which can extend beyond 1 year, the organism will be shed from time to time, and this will be a source of infection for a long period of time. This has been very appropriately described in this individual called Typhoid Mary. Very unfortunate incident, said that she was the carrier, and she carried the organism probably in the gallbladder, and from time to time she started shedding the bacteria, infecting a large amount of people. and therefore she was quarantined for 26 years I think in somewhere in New York, and she died a very lonely death. So this is an extreme case of carriers of Salmonella typhi. Coming to the laboratory diagnosis, in the laboratory diagnosis, the gold standard always is culture of the bacteria. So you have a blood sample because the bacteria invades into the systemic circulation. The organism able to one is able to isolate the bacteria from the blood. At the base of the base laboratory, the sensitivity of the blood culture may not go beyond 40 to 60%. Sometimes, if the blood culture is negative, the patient is still suffering from fever at the end of 3 or 4 weeks, one can do a bone marrow aspiration and do a blood and bone marrow culture, whereas one should be able to identify 90% of the time one should be able to isolate the bacteria. In addition to the blood culture, one can also do urine and stool culture, one can do nucleic acid based PCR from the blood samples, and one can look for the antibody assay. While these three, blood culture, the bone marrow culture, as well as PCR, can be done at any point of time very early in the disease, but the withdrawal test or antibody assay will be only positive probably at the end of 7 to 10 days, as antibody will always arise in the blood a little bit late. So quickly, so one can do blood culture right on the day 1 from the infection up to 3 to 4 weeks, if and one should be able to isolate the organism. The stool can be cultured from the second week onward. Similarly, urine on the third, but stool and urine, the culture sensitivity is little less. As I told you, withdrawal is one of the oldest assay that has been available for almost for last 100 years, looking at the antibody against O and H antigen, specifically as I already mentioned earlier. So what is the taste like? Before that, the organ from the blood culture, one isolate the gram-negative bacteria, non-lactose fermenting bacteria, and do specific test for Bacteriaceae, identify it as a salmonella, then subsequently do serotyping and identify it as a Salmonella typhi or Salmonella paratyphi or paratyphi B. Okay. The important thing about to know is about VI antigen. The VI antigen is normally carried by Salmonella typhi, and antibody to VI antigen suggests that the patient individual probably antibody to VI antigen suggests that probably the individual is a carrier. That means antibody to VI antigen is produced at a very late stage of the disease, probably because there is overstimulation of the VI antigen, and therefore the VI antigen is positive if it is a carrier. But at this moment, when the conjugate vaccine, the typhoid conjugate vaccine is based on the VI antigen. So if you have a large amount of VI antibody in the blood, this individual may also be a vaccine recipient. Okay. So withdrawal test, I already told you that it is based on the O antigen, H antigen. O is the somatic antigen, H is the flagellar antigen of the Salmonella typhi. So based on the O and H antigen, if you have a anti-H O antibody, anti-H antibody more than 100 and 160, 100 and 160 O antibody as well as antibody, this individual is taken as a positive. If it is beyond 1 is to 320, this is definitely positive. If it is less than 1 is to 160, it is taken as a negative. The disadvantage with this with the withdrawal test is that withdrawal test in endemic area, most of us living in endemic area like Delhi will have invariably a large amount of antibody against both the O as well as the it may have a O antibody or H antibody. The other thing you have to also remember that O antibody appears early and it disappears early, whereas H antibody appears late and lasts for longer period of time. So in the Widal test, we do anti-O and anti-H antibody, and a cutoff is 1:160 for both O and H antibody is taken as a positive sample. The other thing you have to understand is that in the endemic area, the individual may have 180, or sometime it may have also 1:160. In this individual, a measurement of a rising titer will be important. So you take another sample at the, say, at a difference of 2 to 4 weeks time, maybe 4 weeks time, and see whether there is a rising level of antibody. If there is a rising level of antibody more than 1:320 both for O and H antigen, then one takes it as a positive. The other assays that have come in the markets are Typhi.MSA, Typhi Rapid IgG. These tests, although commercially available, have varying sensitivity and specificity. As you notice, most of these assays that we describe, other than blood culture as well as nucleic acid assay, these antibody based assays, only antibodies based assays are available. There is no antigen based assay available because the amount of antigen that is present in the blood from time to time, that release from the, say, gallbladder or release from the blood, is very small to be detected by a standard enzyme linked immunosorbent. So as of now, for diagnosis of Widal test, the first is the gold standard is blood culture, followed by nucleic acid assay based assay. Thirdly, we have a Widal test as a supportive test, diagnostic test for and diagnosis of enteric fever. The other tests, as I told you, Typhi dot, Typhi rapid IgG, IgM, Tubex, TF, have although shown initial promise, but as of now they are not very commercially very successful. I already mentioned about the diagnosis of carrier, that if you have VI antibodies, antibodies against the VI antigen, this will suggest this is probably suggest a carrier stage, as it is positive in 80%. But the new vaccine, typhoid conjugate vaccine, is based on the VI antigen. So if you have antibody to VI antigen, this will also mean that this is a vaccine recipient. What is the treatment of Salmonella typhi? This is a gram-negative bacteria. So initially chloramphenicol, amoxicillin, cotrimoxazole, ciprofloxacin, ofloxacin was administered to treat Salmonella typhi. But as you realize that the organism, because of the plasmid, because of the presence of the pathogenicity island and ability to produce a large number of extended spectrum beta-lactamases, the organism has become resistant to chloramphenicol, it has become resistant to amoxicillin, cotrimoxazole, and the fluoroquinolone drug, because it has able to mutate its DNA guides and produce resistance to ciprofloxacin. As of now, ceftriaxone as well as azithromycin, these are the standard for treatment of Salmonella typhi or typhoid fever. But there has been report of third generation cephalosporin resistance as well as azithromycin resistance from different parts of the world. For example, azithromycin resistance and ceftriaxone resistance has been reported from India, from Bangladesh, from South Africa, and so on and so forth. So in this individual, carbapenemase is the drug of choice. So you have it over a period of time, because of the acquisition of drug resistance, one needs to be very, very careful what antibiotic one needs to give. As I told you that this organism, because of the pathogenicity island in the plasmid, it has the ability to mutate its DNA guides, has required resistance to a large number of different bacteria. Therefore, this organism has become a multi-drug resistant bacteria at different parts of the body. So you can see the multi-drug resistant bacteria. This is the endemic zone, the dark brick color area. So all the brick color area, most of the Africa, most of the South Africa, Indian subcontinent, China, and part of the Southeast Asia are have multi-drug resistant salmonella typhi, which are resistant to, as I already told you, the chlorantraniliprole, chloramphenicol, amoxicillin, uh, fluoroquinolone group of drug, as well as some third generation antibiotic, third generation cephalosporin. And as I told you, as far as the azithromycin is concerned, there has been report from Bangladesh and the South Africa of azithromycin resistance. So in this condition, what is the best way to prevent salmonella or typhoid fever? As you right in the middle of the beginning of the talk, we discussed about that it is a water transmitted or fecal-oral route. So it's a safe drinking water, good sense of sanitation or hygiene, health education, and screening of food handlers. These are conditions which are able to help you prevent salmonella typhi infection. As far as the vaccination is concerned, there are three more very effective vaccines available. Live attenuated, the earlier live attenuated TY21A, VI polysaccharide vaccine. Live attenuated TY21 vaccine is actually mutant vaccine, mutant bacteria, Salmonella typhi. This will go inside the intestinal epithelium, multiply maybe a couple of generations, and then it will die and it will lyse, and it will liberate large amount of antigen, and this antigen will induce immunity. So this immunity will produce mostly the local immunity, IgA, as well as cell mediated immune response. But in recent years, VI polysaccharide vaccine has been also used as a vaccine. But the disadvantage of this vaccine is that this vaccine is not effective below 2 years. Children, infants below 2 years of age doesn't have the capacity to process polysaccharide antigen very well. So this is a VI polysaccharide vaccine is not a good choice for children suffering from typhoid fever. So in recent years, an excellent vaccine has been produced against the typhoid fever, which is called typhoid conjugate vaccine. Now the typhoid conjugate vaccine is also based on the VI polysaccharide. This VI polysaccharide of the typhoid vaccine VI polysaccharide typhoid vaccine has been conjugated to tetanus toxoid uh, of the Clostridium tetani, and this has shown very good protective efficacy against Salmonella typhi. And this vaccine can be given in children below 6 months of age, one single dose, and this can be also given in adults up to 45 years. The difference of this particular vaccine with the rest of the vaccine, like the TY21 or only VI polysaccharide vaccine, is that this has improved immunological property. The typhoid conjugate vaccine, like the meningococcal conjugate vaccine or pneumococcal conjugate vaccine, produces long-term immunity. This can be used in younger children, like pneumococcus haemophilus influenzae B conjugate vaccine. This will be given at the age of 6 months. As I already mentioned, that most of the developing country typhoid fever occurs or mortality occurs in children. So this vaccine is given at the age of 6 months, so it can protect occurrence of typhoid fever, and it has a longer duration of protection. Minimum 5 years protection has been seen. At this moment, WHO doesn't recommend booster dose for the typhoid conjugate vaccine. This vaccine has been licensed in India. It's called Type Bar TCV, and this vaccine is prepared by Indian company Bharat Biotech, and it is a WHO pre-qualified vaccine, and it is available worldwide, and it's not very expensive. Indian Academy of Pediatrics immunization guidelines also advises typhoid vaccine TCV 25 mg, single dose of typhoid vaccine, and 6 months onward. A booster dose is not advised at this moment. So we discussed about the typhoid fever. We discussed about uh, the pathogenesis. We discussed about the epidemiology. Lastly, we discussed about the diagnosis. As I told you, blood culture is the gold standard, followed by withdrawal test as well as PCR or nucleic acid based assay in some center. The worrying thing about the treatment is that salmonella typhi, because of the large amount of antibiotic misuse, it has acquired antibiotic resistance to varieties of different antibiotic, including azithromycin, as well as fluoroquinolone group of drug, as well as third generation cephalosporin. So this scenario is very very critical, and one should be very very careful about how one does treat a case of typhoid fever. Lastly, we talked about the vaccine. There are three vaccines available as Salmonella typhi TY21A, based on the live mutant strain of the bacteria. The second is VI polysaccharide vaccine, which is mostly used in the adult. Lastly, and the latest is the typhoid conjugate vaccine, which is basically based on VI polysaccharide conjugated to the tetanus toxoid antigen, and it has a better immune protection, and it is advised as a 0.5 ml injectable to children, and a booster dose is not advised. Lastly, I also want to emphasize that the immunology of the salmonella typhi or typhoid fever is not well understood. The correlates of protection is not at all clear. Antibody to O-somatic antigen, H-flagellar antigen, as well as other salmonella typhi antigen probably as a whole protects the individual. In addition to because part of the time the organism is intracellular inside the macrophages and monocyte or dendritic cell, cell-mediated immune response is probably very very critical. In addition to this, the mucosal epithelium, the paired species or gut-associated lymphoid tissue, the B cell present there produces the large amount of mucosal Ig. So this mucosal IgA also probably plays an important role in the defense of the individual. Thank you very much.